Okay, now the problem is if you have a large sequences, for example, if you have the 100 sequences and 95 residues, right, 2 sequences, one with the 100 residues and with the 95 residues, and if you use all possible alignments, you can have about 55 million possibilities. So, how to get the best alignment? So, the most obvious one is exhaustive searching, but that is not possible because of a lot of combinations. So, in this case we need to develop a smart algorithm, different ways to get this alignment. So, one of these aspect is dynamic programming. How it works? In this method it breaks the problem into a reasonably small ones and do the analysis and finally combine together to get the final answer. For example, if I give this sequence CA, CGA and CGA, first I start with the first one, take the first one C and C. There are 3 different ways you can align the first one. What are 3 different ways? Right, you can make C and C together, or you can put a C and a gap here, and you can put a gap here and C here. Okay, if you do C and C together, then this is aligned. So, you get the score of +1. If you C is taken out, then this is the remaining one. And the second aspect, if you put the gap in the second one and the first one is already aligned, so remaining is CGA, and the second one we did not do anything, so CGA is as it is. Third option, if you put the gap in the first one, right, so we did not use any nucleotide in the first sequence, so it is as it is. Second one includes the C, so you get GA. So, you can continue as it is and then appropriately score, and finally you can add the whole scores and then see what will be the probable alignment. So, in this case there are various routes for a particular sequence, pair of sequences. For example, if you say sequence A, this is sequence A 368 nucleotides, and the sequence B is longer ones. There are different ways to align. For example, if I align this way or this way, here I put together everything on the right side. Second one I give gaps in between, here is some gap and here is a gap. So, which one is the best alignment, this one or this one? First one, if you look into the alignment, you can see because here everything is at the right side and some of them are aligned properly. So, we tell this is a good alignment. Second, also if you see they properly put some cases which are whatever the aligns, they put the aligns right, and the other places they put the gaps. So, which one is a good alignment? So, which one you need to use, right. So, different ways. For example, there are 2 sequences. When you align the sequence, here this is a match because both are same, I will discuss earlier. This is a mismatch because both are different. Here we inserted the gaps here, right, so this is because this is deleted, so deletion gap. Here we inserted the nucleotide, so insertion gap. Okay, now how to align? So, here I put the sequence A here and the sequence B here. There are different ways to align. So, if you make this type of alignment, first one is C, so C and C connected and 3 gaps. So, here if you go on the x directions and then again they aligned the 3 residues 1, 2, 3, right, 1, 2, 3, and then again 2 gaps, here 2 gaps, and finally they aligned, there is the corner. So, take the first one, this is sequence A, this is sequence A. So, here this is sequence B. First we C and C we aligned, so C and C we put it here and they put 3 gaps. So, in this case we moved 3 characters 1, 2, 3, then we have 3 to 4 3 letters 1, 2, 3 they are aligned. So, their corner 1, 2, 3, right, these 2 are here and we have the 2 gaps. So, we go the down vertically down 2 gaps and the last one is here. Okay. So, now if you have this one, okay, this is the path. Now, if you align the sequence in a different way, here we do not care about the matching or mismatching score, just we try to align. So, now the last one is a gap. So, here everything we aligned and then finally you put a gap here, right, here this is a gap. So, now this one, if you put a gap at the first one, so this is a gap and all others are aligned. So, this is another example. If you see one gap is here and here this is aligned, C and G are aligned. So, then again 2 gaps and here again the 3 aligned and then 1 gap, right, and 2 are aligned and 1 gap. So, now there are multiple ways, multiple pathways to align the sequences, these are the various ways. Now, the question is which one we need to choose, which one is the best one, right. So, in this case we give a scoring function, right. For example, if we give a scoring function, if it is 8 match, then we give a score of +8 and with a mismatch we give -5 and gap symbol we give minus 3. For example, if it is match means weight of x comma y equal to 8 if x equal to y, right. If it is a C and then a C, so we can see this match score equal to 8. Mismatch score if x is not equal to y, for example, one is C and another is T, so we can give this score minus 5. Gap symbol we give minus 3, for example, there is C and the gap give minus 3. This is an example. In the reality, if you see, the gap symbol should get more penalty than the mismatch score. So, if you do like this, first if you see C and C match, so score will be 8, and the second one we introduce a gap. So, what will be the next one? 8 minus 3 equal to 5, because 8 here and because the gap minus 3, that is equal to 5. So, again we introduce a gap, so this number will be 5 minus 3 equal to 2, fine. So, the next again we gap minus 1. So, likewise then we add the score T plus T, that is plus score minus 1 plus 8 equal to 7, and then go with this one, this is a mismatch T and A. So, minus 5, this is equal to 2, finally we get the score that is equal to 12. Now, so we want to see there are various numbers you get depending upon this one alignment. So, we have different ways of pathways. So, different pathways will give you different numbers. So, to combine everything, right, Wunsch and Needleman, they brought up an algorithm, a smart way to select which one we have to align. So, for example, if first sequence is A1 A2 AM and the second is B1 B2 Bn, so we can align different ways you can align. So, if they give proper initialization and they give a score, that is Sij. If you have one nucleotide and if you want to make the alignment compared with the previous ones, either we can give gap for insertion or for deletion, or you can compare these two nucleotides giving the match score or mismatch score. So, he used these values, right, and take the maximum. First way is whether it is insertion or deletion, right, or this is a matching score or mismatching score. You weight depending upon A and B. If it is a match, we give the match score. If it is mismatch, we give mismatch score. Otherwise, we give the gap score. Okay, we give the maximum values, because why he gave the maximum values?
To maximize the score.
Because we want to have the best alignment, so he wants to maximize the score. So, with all the three conditions, he took the maximum of all the three options. So, here take the value from left and add a gap penalty along left axis, or take the value from the above and the gap penalty along the y axis, or take the diagonal and take the weight, whether this is match or mismatch. So, how to do that? So, first we give the gap symbol minus 3, right. So, we initialize first. So, we give minus 3, if we completely gap, then we give this minus 24, right, up to the both ways. Okay. Now, first case is first we have to align. So, which one is the first number? So, three options for this one. What are three options?
0,
minus.
One we can come from here, or we can come from here, or from left to right. Here we introduce gap, here we introduce gap, and here we introduce the alignment either match or mismatch. So, option 1 yes 0 0 came from here, weight of A1 comma B1. So, here if you see A, what is A1? C, what is B1? C, the match. So, match score equal to 8. So, we put 0 plus 8 is equal to 8. Then option 2 we go from S11 that is equal to S01 plus weight of A1 comma gap. If you do this, this minus 3 relative minus 3, so put another minus 3, so this will be minus 6. In option 3 we go this 1 comma 0 and with the gap in the first term and the B1. So, if you see this one, this gap is minus 3, so again it is minus 3, this will minus 6. Now, compare these 3, one is 8, one is minus 6, one is minus 6, which one is the maximum? 8. So, with the maximum value of 8 here. So, what is the value here? What are the possibilities? The numbers from here, what is the number? Minus 8, this way minus 9, and this way this 5. So, 5 is the maximum. So, you put 5 here. So, now this 5. So, what is the number here? These are the options minus 8 plus 5 and minus 9, right. So, the maximum is 5. So, you put 5 here. fine. This one quickly, 5 minus 2 equal to 3, here equal to 5 minus 3 equal to 2, 5 minus 3 equal to 2, and here 8 minus 5 equal to 3 because of mismatch. So, this is the maximum, so we put 3 here. So, finally, we fill the matrix. So, finally, we get the optimum score of 14. So, now the question is how to go back, because we get the maximum value. From the highest value we go back, because there are 3 possibilities, 3 directions, and see which one is the best, you go back and then finally make the alignment, right. This is the 14 is the highest one, then go back among the 3, this is the 6 and then this 9 and 1, 6 minus 2 and 3, and here we see 8. So, now here we have one, we introduce one gap here. So, this gap is here, T and T are aligned, right, and then if you see all others are aligned, C C, T G, T G, A A and A T C C, right. So, we aligned all other sequences. So, you put the number, so finally we get total of 14. So, now if you have 2 sequences, we check the 2 sequences and there are 3 possibilities to fill the matrix, and take the maximum number and fill the matrix and trace back to see the root, and finally you can make this alignment. So, here there are 2 types of alignment. One alignment is the global alignment, as I discussed in the previous classes. Here it considers everything, all the residues are aligned, and in some cases we align only some particular regions. Okay, this is called the local alignment. Okay, for why we need local alignment? For short repeats, or some cases there are small motifs which are important for any specific functions, like TA box binding protein. So, Tata box is important for the binding. So, if you want to see the small repeats or small motifs, we can get from this local alignment, right. For example, if this is the sequence A and sequence B, some cases we consider all the sequence together. So, for the global alignment, here gaps are also counted, and here you can see we ignore the gaps and then see only the other regions, right, for local alignment. And also another alignment called semi global alignment, this will tell you how we can align the different sequences with respect to gaps internal or the outside. Some case if you see the gaps outside, these terminal gaps are usually the result of incomplete data acquisition and may not have any biological significance. In this case you can remove these gaps, and if you remove the terminal gaps, then we align only the sequences which is in between. So, this type of alignment we call as semi global alignment. Okay, how to do this alignment? For example, if you get 2 sequences, okay, this is sequence 1 and this sequence 2, we can make different types of alignments. For example, you can make this type of alignment. If you look like a global alignment, it is a kind of only bad alignment, because one gap here and some gaps here and the residues are not properly aligned. But if you look into details, right, it will give you the matching region called TATA. So, then we how to extract this type of information. So, in this case Smith and Waterman, they proposed another option. So, currently there are 3 options, right. Wunsch and Edelman proposed 3 options. What are 3 options? Match or mismatch, deletions, insertions, right. They take the maximum of these 3 conditions. So, they proposed on the 4th option, so the value of 0, they do not want to have any negative values, right, they want to have the positive values. If it goes a negative, just remove it, because we do not have to include in the alignment. So, they include the alignment only the positive values. So, now the condition is the maximum of these 4 options. One is Si minus 1, Sj minus 1 diagonal, right, go through diagonal lines. So, with A and B with respect to match and mismatch. The second one is the gap, third is also gap insertion and deletion, and the last option they give 0. So, if we have this same sequence, I give a sequence, first initialization because there is no negative values, so put as 0, right. What will come here? 8, right, this is equal to 8, because C and C, this is 8, this is a match, and here this is a gap is minus 3 and the other gap is insertion minus 3. So, this is option is 4, this is 0. So, the maximum of this among this 4 is 8. So, I put 8, right. What is the next one? What will come here?
Next one 5. 5 plus a
13.
13,
Yes.
right, 13, right. So, now we fairly completed this matrix, right. So, now the maximum score is 18, right. So, now go back with this 18, so go with this 10, here this again among these 3 is 13, so here this is 5, this is 8 and then 0, so they stop here. So, make here, so this is the 18 T and T match, right. Here then we use this gap, so A, then C and C are matching, right, then another gap, so the A and D are matching, so it is equal to 18, right. So, I will give you another example. So, here these are two sequences, this is sequence 1, this is 2 and how to make the alignment. So, for simplicity I put the match score equal to 1, mismatch score equal to -1 and gap symbol equal to -1. So, what is the value?
Minus
0, that is 0, because in this is a maximum of 4 conditions, right, the maximum is 0, right, we do not have any negative values, right. So, it is 0. So, if you make the alignment, finally we get these numbers here equal to 1, this equal to 2 and this equal to 3 and this equal to 4. So, now this is maximum, right. So, now we trace back 4, 3, 2, 1. So, finally if you see the alignment, this is A and A, okay, this A and this A, right, A and A aligned, and the T and T, this T and this T aligned, and this A and this A aligned, and this T and this T aligned, after that is 0. So, we get this motif between these two sequences, right, this is a specific motif for the DNA binding proteins, right. So, we could find any specific motifs or any patterns, right, in any alignments. So, this is the applications for this local alignment. So, there are two questions. So, we can use these two different algorithms, right, to align the sequences 1 and the sequence 2 either with the local alignment or with the global alignment. So, so far what did we discuss today?
Algorithms for alignment.
Yeah, the algorithms for alignment, right. So, how to, if we have two sequences, right. So, different ways to align, right. So, what is dynamic programming?
So we divide the alignment process into smaller parts.
Small parts, yes,
We'll align small fragment will align and
then finally merge together to form the complete alignment, right. Right. So, what are the two different algorithms we discussed today?
Needleman-Wunsch algorithm
Wunsch-Needleman algorithm
and Smith and
Waterman algorithm. Wunsch-Needleman algorithm is for which type of alignment? Global alignment, right. What is the condition used in Wunsch-Needleman algorithm?
substitution means
Yeah, there is a maximum of
Substitution score.
substitution.
3 different conditions, right. So, either substitution score or the insertion or the deletion. In the case of local alignment,
Yeah, the fourth condition.
fourth condition, what is the fourth condition?
If value is
That is 0, right. So, now we can get the values. So, till now we use some numbers for the match score or mismatch score or the graph, we use some numbers, right. In the beginning of the lecture we discussed about some matrices. So, what are the matrices we discussed? PAM matrix and the blossom matrix. So, what are the characteristics of PAM matrix?
it considers mutations.
It considers mutations, right. So, depending upon the type of mutations or based on this charge or the actually it depends on the actual mutability rate, right. In the actual cases depending on the mutability rate, so that derive the matrix. So, accordingly you can use the numbers available in the blossom matrix or the PAM matrix to give the weightage, either the match or the mismatch, you give the weightage, then you can give the probable alignment, right. This is what they use in the BLAST when they implement the algorithm. So, next class we will discuss about the software for aligning sequences. How now we discuss about how BLAST works. Now, I will tell you how they take two sequences and how they align the sequences and what is the score they give, how they evaluate the score between two different alignments. For example, if you have one sequence which is aligned with 3-4 different sequences, which one has the highest score, which one has the most probable alignment, right. So, they give the alignment scores, they try to optimize this alignment, right. We will discuss in the next class. Thank you for your kind attention.